Welcome to part 2 of section 1.4. So in this video we are going to be looking at microscopes and two different types of laboratory techniques. I'll touch briefly on the safety in the laboratory, but since you have already looked over that information, it is just there for you to take a peek at. Alright, so microscopes. Again, microscopes are devices that are going to produce magnified images of a structure that's too small for us to see. And we're going to use microscopes here in biology, especially when we look at the cellular level. There's two different types that you're going to need to know, and those are light microscopes and electron microscopes. So the light microscopes use light rays and lenses to magnify. Often we can get 1,000 times the magnification of our unaided eye. Again, light microscopes use light ray and lenses. We also have a compound light microscope, which is 2 lenses. So when we think of compound 2 or more, and that's what a compound light microscope is, 2 or more lenses. When we look at the electron microscope, this works by focusing beams of electrons. So electron microscope, electrons. Light microscope uses light and lenses. These are even more high-powered, and they're 1,000 times more detailed than light microscopes. So electron microscopes allow us to see a lot more than even a light microscope. So two lab techniques that we're going to look at is a cell culture and cell fractionation. So a cell culture is really growing cells. Let me do so we're actually growing cells. And what generally happens is you take one cell, plus nutrient solution, and you get more cells. And again, the purpose of this is to really, we can use cell cultures to test cell responses, study interactions between cells, and actually pick out specific cells within each to study them a little bit more. So a cell culture is really growing cells, so we can use them. Cell fractionation, on the other hand, if we analyze what is a fraction? A fraction is really a portion of the whole, right? Portion of the whole. So what do you think we're trying to do with cell fractionation? We are trying to get a portion of the cell. So what they really do in this one is it's this goal to separate into parts. They put it in this handy dandy little blender, and they mix it up. And then what happens is they take that goop and pour it into a test tube. We got it in there. And they shake it up using a centrifuge, and then we get these nice little layers. So we might have something down here, something down here, and something here. So again, they can pull out different layers and actually study those. So that's something that we also use is if we're looking at a specific part of the cell, we'll use cell fractionation. Lab safety. Again, this is stuff that you have already seen, you already signed off of, that type of thing. The biggest part with lab safety is really number 4. Okay, read the lab instruction carefully. Read lab instructions carefully. Many questions you might have are answered in your land. How about do not skip steps and follow the direction in the order they're given. Also listen if I give any directions as well. So listen to the instructions as well. I might have instructions that are a little bit different than what's in the lab manual, so you want to make sure that you have those. And make that a little bit smaller. You can type in. Alright, so I'm going to just show you the next slide as well, and that is it for this. So this is the last piece of that. Again, remember, wear appropriate clothing. If there's something different that I need you to wear, I will go ahead and do that. Otherwise, this ends section 1.4.